Today I'll be presenting on Ofloxacin, a synthetic fluoroquinolone antibacterial agent that functions by inhibiting bacterial DNA rest, thereby halting the DNA replication. The name Ofloxacin comes from two parts. Oxa refers to a carbon atom being replaced by an oxygen atom, while Ofloxacin is a suffix commonly used for fluoroquinolone antibiotics. Ofloxacin is a part of fluoroquinolone class developed in 1980s by Daiichi Sankyo Co. in Japan. It's chemically modified from nalidixic acid and is unique because it's racemic mixture, meaning it contains both left and right-handed molecules. However, although levofloxacin is active. The molecular formula of ofloxacin is C18H20FN3O4. Ofloxacin appears as a bright to pale yellow crystalline powder. In terms of solubility, it's slightly soluble in water, alcohol, and methyl alcohol, and sparingly soluble in chloroform. An important point for handling is its decomposition, which emits toxic fumes like hydrogen fluoride and nitrogen oxide. In aqueous solution, its pH range from 3.8 to 5.5, and it has a half-life of around 5 to 7 hours in healthy adult, which is useful for dosing schedules. Look at how it works. Ofloxacin mainly targets two bacterial enzymes, DNA GRAS, also known as topoisomerase 2 and topoisomerase 4. For DNA GRAS, it introduces negative supercoils into DNA, which is essential for replication. Ofloxacin binds to the DNA GRAS DNA complex and prevents the resettling of DNA strands after they are cut, which causing cell death. This is particularly effective against bacteria like E. coli. As for topoisomerase 4, it's responsible for separating interlinked daughter DNA molecules after replication. Ofloxacin stabilizes the DNA topoisomerase complex, preventing chromosome segregations. Therefore, ofloxacin effectively disrupts replication in Staphylococcus aureus by targeting topoisomerase 4. There are four mechanisms, and the first one is target site mutation. Changes in the genes encoding DNA GRAS and topoisomerase reduce drug binding. For example, Salmonella typhi uses this mechanism. This bacterium can acquire resistance via point mutation, making fluoroquinolone less effective. Next, efflux pumps. Bacteria like Pseudomonas aeruginosa overexpress efflux pump that actively push the drug out of the cells. This organism has a well-known efflux system called MAXPQ-OP-ME to act resistant against fluoroquinolones. Other than that, reduce permeability. Some bacteria like Acinetobacter baumannii produce fewer porins, reducing drug entry. This pathogen may express fewer porins, making it harder for ofloxacin to enter and act on the cells. Lastly, plasmid-mediated resistance. Genes on plasmid encode protective proteins like QNR in E. coli, which share the energy rest from ofloxacin. In terms of cost and availability of ofloxacin, the oral tablets cost about RM21.90 per strip. For eye infection, 0.5% eye drop formulation called Zanaflox, which costs RM22.20 for a 5 ml bottle. There's also an otic drop called Efficacin, priced at RM23 for 5 ml. For more serious infection that need hospital treatment, there's a intravenous infusion, which costs about RM21.84. That's all for me. Thank you.